What if the true story of human origin wasn't just about where we came from, but about what we became when we converged? In the heart of East Africa, where the savannas rise into the highlands and the forests thicken toward the equator, Uganda holds a genetic mirror unlike any other. This land isn't just where people settled, it's where civilizations collided, fused, and transformed. Before kingdoms, before maps, before memory itself, people were already moving through this terrain. Some came with cattle, some with stone tools, some with fire in their stories and bone in their hands. And they didn't just pass through, they stayed. They became one another. Geneticists who've studied the people here don't speak in myths. They speak in sequences, in Y chromosomes, in empty DNA, in clusters and clines. When they looked into the DNA of Nilotic-speaking Ugandans, they found something that textbooks never mentioned, a Y DNA variant so rare, so localized, it could only have arisen from deep-rooted lineage. B2A1B, a genetic fingerprint invisible in neighboring Bantu populations. That single marker is not just a genetic fact. It's an ancient voice saying, "We were here. We took a different road." But these weren't isolated roads. The Bantu, whose roots stretch westward into Central Africa, arrived with agriculture in their bones. Their expansion was not a wave of conquest, but of coexistence. Their genes blended with local populations, but they also preserved lineages distinct to their journey. In modern Uganda, Bantu-speaking groups like the Baganda and Basoga carry maternal lineages that intertwine with Nilotic groups, a sign of shared lives and complex unions that no history book recorded. Then, deeper in the genetic strata, researchers found the Batwa, rainforest hunter-gatherers whose DNA carries an echo from the Pleistocene. These were not merely short-statured people in the forest. They were the descendants of a separate evolutionary path. Studies of their growth genes revealed distinctive patterns in hormone signaling, bone structure, and development. This wasn't cultural adaptation. This was evolution answering to a different world. Dense canopy, endless rain, a rhythm of life that required something entirely different from what agriculture would later demand. The more genomes were mapped, the stranger the story became. Mitochondrial haplogroups usually found in parts of the Near East, K1A, R0A1A, N1A1A3, began appearing in Ugandan populations. So did Y chromosome haplogroups like R1B and H. These weren't recent arrivals. They had been here for millennia. Ancient DNA analysis and statistical modeling confirmed it. There had been back migrations into Africa, movements from the Levant or even further, returning not as conquerors, but as fragments of long-forgotten trade, pastoralist spread, or climatic refuge. Their descendants still walk the hills of Uganda, unaware they carry genes shaped by Neanderthal ancestry picked up on those prehistoric roads. When scientists used principal component analysis to map the genetic structure of Ugandans, they expected some regional variation. What they found was something else entirely. Not one homogeneous cluster, but dozens. In southwestern Uganda alone, 52 distinct population clusters emerged, each tied not just to geography, but to language, to culture, to a unique past encoded in the blood. The Baganda, the Bakiga, the Lugbara, all separate, all distinct, yet all tied together through threads that only DNA could trace. In Uganda, migration wasn't a one-time event. It was a way of life. Waves of humanity washing over one another, never erasing, only adding. Generation after generation. The result is a population whose diversity cannot be simplified. A living archive, storing the routes our ancestors walked and the choices they made. This is not a story with a clear beginning. It's a tapestry of intersections, a convergence of pastoralist and farmer, hunter and herder, migrant and native. A place where migrations didn't merely pass. They collided. And from that collision came one of the most complex genetic legacies on Earth. There are places where history is carved in stone. And then there are places where it's written in blood. In the genomes of ordinary Ugandans, farmers, teachers, herders, children, scientists uncovered something that silenced their assumptions. It began with a routine genome sequencing project. Nothing revolutionary was expected. But as the data poured in, patterns emerged that no global reference could explain. Over 41 million genetic variants, nearly one-third of them absent from every international database. Not rare. Unknown. That wasn't just a surprise. It was a disruption. For decades, global genomics leaned westward. Databases like NAMAD and the 1000 Genomes Project, though powerful, had an enormous blind spot: Africa. Uganda, with its vast ethnic diversity and layered ancestry, had simply not been part of the equation. And so, the scientists kept missing something. The story wasn't incomplete, it was deeply distorted. Then came the Uganda Genome Resource, 6,400 individuals sequenced, with nearly 2,000 full genomes mapped in unprecedented detail. These weren't just numbers. They were revelations. Genetic variants unique to Uganda surfaced, variations that affect how bodies respond to medications, how diseases take hold, how traits are passed from one generation to the next. Some of these variants influence cholesterol levels. Others regulate blood pressure or immune responses. Many of them would have been invisible in European-centric studies. And so, for years, their effects went misunderstood, or worse, ignored. Even the way genes are linked in Ugandan DNA looks different. Where Western genomes show clear, consistent patterns, what geneticists call high-linkage disequilibrium, Ugandan genomes show more fragmentation, more independence between genetic markers. It's a fingerprint of something older, more complex. It suggests not just age, but diversity shaped over tens of thousands of years. These aren't echoes of one migration. These are the signatures of many, colliding, diverging, recombining across the millennia. The diversity isn't just high, it's explosive. Researchers documented 52 distinct subpopulations in a single region of Uganda. Each one carried clusters of genetic traits, many still not fully understood. To a trained eye, these weren't just population differences. They were potential breakthroughs. Traits linked to blood sugar regulation, genetic factors involved in kidney disease, mutations that could unlock treatments for conditions like sickle cell, diabetes, and even autoimmune disorders. But these insights come with a warning, because without African DNA in global datasets, precision medicine risks becoming a kind of medical colonialism, offering solutions for a few while ignoring the many. Ugandan genomes highlight the danger in generalizing biology. What works in one region may fail catastrophically in another, and what seems benign in a Western genome might carry serious consequences when translated to African populations. That's why this isn't just a scientific story. It's a human one, because inside these genomes are real people, communities whose lives have been shaped by evolutionary pressures unique to this land. Malaria, drought, high-altitude exposure, viral epidemics, each left an imprint on the DNA. And until now, those imprints were missing from the world's medical map. But Uganda is changing that. Local researchers, many trained through programs like H3 Africa, are not just participating in the genomic revolution, they're leading it. Studies from Makerere University are informing disease modeling worldwide. Data from the Uganda Genome Resource is now part of the International Haplotype Reference Consortium. And with projects like Kidney Gen Africa and DEEP pushing forward, Uganda's genomic voice is no longer silent. This is not a story of catching up. It's a story of redirection. Because the code in Ugandan DNA isn't just an outlier, it's a compass. It points to where science needs to go next. Not because Uganda is a mystery, but because it is an origin. A place where the diversity of human life was never narrowed, never erased, only layered. And that's why the world couldn't see it before. It wasn't too hidden. It was too complex. In Uganda, evolution is not a theory written in textbooks. It's a story engraved into bodies, passed through generations, like a whispered warning from the past. This is a place where survival has never been abstract. The land, lush and unforgiving, has tested its people in ways few regions ever could. And those who remained, who endured, carry evidence of that battle in every cell. It begins with malaria, a disease that has haunted the tropics for thousands of years, shaping not only societies, but genomes. One mutation, just one shift in hemoglobin structure, offered a chance at survival. The sickle cell trait. Inheriting a single copy meant resistance to the deadliest forms of malaria. Inheriting two could be fatal. It was a gamble written into life itself, one that nature made again and again. In parts of Uganda, Over 20% of people carry this mutation. It's not a flaw. It's a genetic shield, hammered into place by centuries of exposure. And malaria was only the beginning. Another layer of defense came in the form of G6PD deficiency, a mutation that changes how red blood cells respond to stress. Carriers can't tolerate certain foods or medicines, but they're more likely to survive a malarial infection. It's an evolutionary trade-off: vulnerability in one area, strength in another. In many Ugandan communities, 1 in 5 people live with this adaptation. They may never know it, but their blood remembers. Then there's the Duffy-negative blood group, a genetic variant that nearly erases one species of malaria from the equation entirely. In much of the world, Plasmodium vivax infects millions. But in Uganda, where most people lack the Duffy antigen, the parasite can't even enter the red cell. It's an elegant lockout, shaped not by medicine, but by time. This battle between parasite and host didn't stop at malaria. When HIV emerged in the late 20th century, Uganda was one of the first countries to confront its devastating spread. But even here, the genome responded. Certain HLA class I alleles, particularly B57 and B5801, are more common in East African populations than elsewhere. Individuals with these variants often suppress HIV naturally, sometimes for years, without antiretroviral treatment. They're known as elite controllers, and their resistance isn't just powerful, it's informative. Studying these people has led scientists to reconsider how immunity functions, not universally, but population by population. Behind that immunity lies another genetic layer: KIR genes, the complex switches that regulate how natural killer cells destroy infected cells. In Ugandans, the diversity of KIR gene combinations is among the highest on Earth. It's not randomness. It's a reservoir of flexibility, a hidden weapon against viral threats that keeps the immune system unpredictable, and therefore more effective. The pattern repeats. In regions of eastern Uganda, where sleeping sickness still appears, scientists found that the APOL1 gene, normally associated with kidney disease, also provides resistance to trypanosome parasites. The same mutation that protects from one death sentence may predispose people to another. Evolution didn't promise perfection, only adaptation, and these adaptations are still evolving. They're not frozen in ancient history. They're changing with each generation, responding to modern pressures, new diseases, shifting environments, the spread of antibiotics and vaccines. Uganda's genome is not a relic. It's an ongoing negotiation between life and death, resilience and risk. These mutations don't exist in isolation. They're part of a larger pattern, a landscape where every trait has a backstory. And every backstory is rooted in environment. Rainforest, swamp, highland, savanna. Each ecosystem applies its own rules, and human DNA follows suit. This is why there is no single African genome. There are thousands of microadaptations, scattered across the continent like beads on a thread. And in Uganda, many of them shine brightest. What looks like diversity from the outside is strategy on the inside. Uganda's people don't just represent a branch of the human family tree. They are one of its testing grounds, a place where the fiercest challenges forge the most intricate defenses. And for those paying attention, every one of those defenses tells a story of danger survived, of choices made, of life held onto cell by cell. Beneath the tangled canopy of Uganda's rainforests, a people live whose very bodies defy mainstream assumptions about human biology. The Batwa, often reduced in modern narratives to the term pygmies, carry within them a truth far more intricate than just height. To outsiders, their short stature may appear as an anomaly, but to geneticists, it is a code, a reflection of deep evolutionary intelligence shaped by thousands of years in one of Earth's most demanding environments. In a world where agriculture defined success and expansion, the Batwa moved differently. Their survival depended not on fields or flocks, but on soundless movement, on intimate knowledge of plant cycles, on the rhythm of the forest itself. Over time, their physiology began to reflect the pressures of that setting, not as limitation, but as optimization. When scientists compared Batwa genomes to those of their taller, agrarian neighbors, they didn't just find size differences. They uncovered genomic divergences in key pathways that regulate growth. Specific alterations in the genes responsible for skeletal development, bone formation, and growth hormone receptors pointed to a deliberate biological shift, not an accident of nutrition or isolation. These weren't malnourished farmers. They were forest specialists. And the deeper they looked, the clearer it became. This wasn't a singular phenomenon. Across Africa, groups who've lived in forested regions for millennia show similar height patterns, from the Aka in Central Africa to the Mbuti in the Congo. But in Uganda, the Batwa present one of the clearest, best-preserved cases of adaptive short stature. Their genetics carry a record of life in a dense, humid, low-light world where smaller body size may have meant better heat regulation, reduced caloric demand, and a stealthier presence among thick undergrowth. Their biology reflects that of people who learned how to disappear, not socially, but ecologically. Yet while their genetics speak of adaptation, their recent history tells a different story, one of displacement. As forests were cleared and conservation laws pushed indigenous groups from their ancestral land, the Batwa lost access to the environment that shaped them. But their DNA didn't forget. Even as their lives changed, the genomic echoes of their ancient ecological niche remained. This is where genetics transcends theory, because inside the Batwa genome is proof of human flexibility, the ability to adapt not just culturally, but physically across generations. And it challenges one of modern science's most damaging assumptions, that there is a standard human body. There isn't. There never was. Each environment writes its own rules, and those rules get embedded in the people who survive them. In the case of the Batwa, that survival came at a cost. Today, many live marginalized on the fringes of Ugandan society. But when you trace their lineage through mitochondrial markers, you find maternal lines that extend farther back than most neighboring groups. They are not new. They are not lost. They are ancient. And the traits they carry are not remnants. They are blueprints. For scientists studying evolutionary biology, the Batwa offer something rare, a living population whose physical traits directly correspond to their ecological past. Their genome isn't abstract. It's narrative. It tells of adaptation without conquest, of strength and subtlety, of survival through refinement rather than expansion. The forest shaped them, not just in myth or memory, but in measurable biological ways. And if we listen, their bodies still speak, not just of who they are, but of who humans can become when we live close to the land, when we adapt to it instead of reshaping it. In every inch of stature, there is strategy. In every gene, history. For generations, the story of humanity was told as a one-way journey, out of Africa, across the globe, away from origin. But buried deep in the genomes of Ugandan communities is evidence that the path wasn't linear. It circled back. Geneticists didn't expect to find traces of Neanderthal ancestry in the heart of East Africa. That legacy was thought to belong to Europeans and Asians, those who lived far beyond the bounds of ancient Africa. But the data said otherwise. Subtle fragments of archaic DNA, remnants of ancient interbreeding events, were found embedded in Ugandan genomes. And they didn't arrive recently. Their roots stretched thousands of years into the past. It wasn't just Neanderthal signals. Entire haplogroups with Eurasian origin, like R1b, K1a, HV1, and W8, appeared in Ugandan maternal and paternal lines. Their presence wasn't widespread, but it was persistent. These were not outliers. They were threads. And when researchers traced those threads through time, they pointed back to something rarely acknowledged: return migrations. Thousands of years after humans left Africa, small groups came back. Some through the Levant. Others likely followed pastoralist routes through North Africa and into the Horn. They carried new genes, new technologies, and new connections to the world beyond. They didn't conquer. They merged. Quietly, gradually, over generations, their DNA blending into the genomic fabric of East Africa. Statistical models like Mulder and Globetrotter dated these admixture events with surprising precision. Some occurred over 4 millennia ago, others as recently as 10 generations back. And in all of them, Eurasian ancestry didn't flood the population. It laced through it, like a whisper in a crowded room. The implications shook the foundations of migration theory. These weren't colonial echoes or slave trade remnants. They predated written language. They belonged to a time when borders didn't exist, when humans moved with climate and food, not flags. And they changed how we define identity. Because if Eurasian genes flowed into Uganda thousands of years ago, then Uganda is not a genetic endpoint. It's a loop, a meeting place. That loop complicates everything. It means modern populations in Uganda are living proof that ancestry isn't a straight line. It's recursive, interwoven. When Ugandan genomes show signals of both ancient African hunter-gatherers and Levantine pastoralists, they aren't contradicting history. They're completing it. This intermingling is visible in mitochondrial lineages passed from mother to daughter, lineages that trace both local continuity and foreign arrival. And it's seen in paternal markers that suggest an ancient Eurasian male presence, likely absorbed into the local structure through trade, alliance, or shared survival. What's most striking is that these foreign genetic elements didn't erase local identities. They were absorbed without dominance. That's rare. In many parts of the world, gene flow came through violence, displacement, collapse. But here, the traces suggest cohabitation, not conquest. Ugandan diversity didn't emerge by accident. It is the result of countless intersections, of peoples who didn't just pass through each other's lands, but each other's bloodlines. The forest dwellers, the cattle herders, the cultivators from the west, the migrants from the north, and yes, even those from across the seas. For every step outward humanity has taken, there's always been a path back inward. The return to Africa didn't just happen. It mattered. And it left a mark still carried in the cells of those who walked the hills, fished the lakes, and raised their children beneath Ugandan skies. In the world of genetics, power has long flowed in one direction, from labs in the West, through data pipelines, toward global conclusions. But a shift is underway, and its epicenter is Uganda. For decades, African DNA was underrepresented in global genomic research. Studies claiming to explain the human genome barely included the continent that birthed it. The consequence? Misdiagnosis, overlooked mutations, treatments built on incomplete models. But then came a breakthrough that didn't just fill a gap, it redefined the map. The Uganda Genome Resource, or UGR, launched with a goal as ambitious as it was overdue. Sequenced thousands of Ugandan individuals, analyzed not just their ancestry, but their health, and build one of the most detailed genomic databases ever assembled on the African continent. With over 6,400 participants and nearly 2,000 complete genome sequences, the UGR didn't simply produce data. It generated context, clarity, and confidence in a landscape previously reduced to genetic footnotes. And this wasn't a foreign project. It was designed, led, and executed by African scientists. Experts based in Kampala, Gulu, and Bale, not London or Boston, analyzed the data. They asked different questions, and so they uncovered different answers. Among the discoveries were genetic markers that impact traits like lipid levels, blood pressure, and insulin sensitivity. Traits that behave differently in African populations than they do in European cohorts. These aren't just curiosities. They're essential to building accurate medical diagnostics and treatment strategies. Without this data, modern medicine risks failing those it was never truly designed for. And the ripple effects go beyond Uganda's borders. Global initiatives like H3 Africa, supported by the African Union and international funders, began channeling resources into locally led genomic research. Biobanks were built, sequencing capacity expanded, training programs multiplied. What started as an effort to document genetic variation became a movement to claim scientific sovereignty. Projects like KidneyGen Africa now aim to analyze 110,000 African genomes to understand why diseases like kidney failure hit African populations harder, and often younger, than anywhere else. Ugandan researchers are among the key architects of this effort, working not just with data, but with patients, families, and communities. In parallel, the DEEP initiative explores the intersections of genes and environment, studying how stress, pollution, nutrition, and trauma leave marks not just on lives, but on the genome itself. Epigenetics is no longer a Western frontier. It's being reshaped in real time by African data. This is more than science. It's reclamation. Because for centuries, Africa's biological resources were extracted: blood, bones, land, labor. Genomics was no exception. Samples were taken, knowledge exported, credit assigned elsewhere. But now, Uganda is not just contributing to science. It's steering it. The UGR has already joined the Global Haplotype Reference Consortium, providing a foundational dataset for researchers around the world. But unlike the past, this collaboration flows in both directions. Ugandan scientists co-author the papers. They set the terms of access. They decide which stories get told. And those stories are changing how we understand everything from disease to ancestry to evolution. No longer is African data used to support someone else's theory. It's being used to build Africa's own. What's unfolding in Uganda is not a catch-up. It's a leap forward, a genomic renaissance, born from necessity, powered by purpose. In classrooms, field clinics, mobile labs, and high-tech sequencing centers, a new generation is being trained to study African DNA with African tools for African futures, not as subjects, as scientists. In a country woven with over 50 ethnic groups, there's one whose presence feels older than borders, older than farming, older even than language as we know it. The Batwa, known for generations as forest dwellers, they once lived in symbiosis with the towering trees and shadowed paths of southwestern Uganda. But their legacy goes beyond tradition. It is inscribed in their DNA, a record of survival in one of Earth's most demanding ecological theaters. Scientists looking into Batwa genomes were not searching for legends. They were searching for patterns, clues buried in nucleotide chains that could explain how a people adapted so precisely to the rainforest. What they found were not merely traces of isolation or cultural difference. They found a genetic architecture, finely tuned to life, beneath the canopy. Across the globe, height is influenced by thousands of genes, some common, others rare. In the Batwa, researchers detected distinctive signatures in growth hormone signaling and bone metabolism pathways that diverged sharply from neighboring farming populations. These were not anomalies. They were consistent, measurable differences. Evolution sculpted their bodies to suit an environment where agility mattered more than size, where navigating thick undergrowth required precision, not reach. But what made these findings even more compelling was their uniqueness. The Batwa's genomic profile showed signs of ancient lineage separation, a divergence so deep it suggested their ancestors had remained genetically distinct for thousands of years. Their adaptation wasn't a result of recent environmental change. It was the outcome of deep time, an uninterrupted existence within the forest ecosystem. And the clues weren't limited to height. When cross-referenced with other rainforest hunter-gatherer populations, the Baka in Cameroon, the Mbuti in the Congo, the Batwa showed some overlap, but also notable differences. Their adaptations were not carbon copies. They were locally specific, tailored by the pressures of Uganda's particular terrain, rainfall patterns, biodiversity, and ecological constraints. In other words, the forest didn't just select for small stature. It shaped the Batwa in ways no other environment could replicate. Yet this biological mastery of environment stands in tragic contrast with their recent history. In the last century, conservation projects redefined their ancestral lands as protected zones, pushing the Batwa to the margins of society. They lost access to the ecosystems that had sustained their way of life for generations. In many areas, they were displaced without compensation, left without legal recognition. But while the world ignored their rights, their genome preserved their truth. This is why the Batwa matter, not just as an endangered culture, but as an evolutionary archive. They hold answers to questions that modern science is only beginning to ask. How do humans respond to extreme ecological niches? What happens when adaptation occurs outside of agriculture, beyond domestication, in harmony with rather than in dominance over nature? Their genome tells us that survival isn't always about conquest. Sometimes it's about listening. The Batwa's biology, like their history, has been misunderstood, reduced to stereotype, stripped of nuance. But when viewed through the lens of genetics, a far richer story unfolds. One in which body size is not a limitation but an optimization. One in which marginalization hides a profound legacy of human adaptation. and one in which Uganda's rainforests echo with a voice few have bothered to hear. That voice is not gone. It speaks through sequences, through markers, through generations. It speaks of resilience without aggression, of ancestry without borders. And it reminds us that within every overlooked population, there may lie an answer not just to who we are, but to how we have endured,